Then write your name and roll number in the chat box. Just write your name and roll number in the chat box. 43. Those are not mentioned. All of you seen my share my screen?
Yes, sir.
Yes, sir.
Yes, sir.
So please stop your camera. And today we discuss about the enzymes in the biochemistry, because enzyme is one of the most important chemical, or it is a biocatalyst which is essential for the metabolism. All the metabolism, all the digestive process can be done by the with the help of the enzymes. The enzymes is such a substances which is so faster, just it present in the reaction vessel and increase the rate of reaction, that is called the catalyst. But enzyme is called the biocatalyst because these catalysis occurs in the biological process. So enzyme is a biocatalyst and gives how much faster the rate of reaction. Gives one example. Suppose you take one piece of chicken in the concentrated hydrochloric acid, it will be digested within 8 to 12 hours. But when this chicken taken kept into the dilute HCl and you use the pepsin enzymes, within 2 to 3 hours it will be digested. So this is so faster the rate of reaction in presence of enzymes, and obviously along with the other substances like coenzymes that or cofactor which help for the enzymatic action. A piece of chicken which is digested in the presence of concentrated hydrochloric acid, around 8 to 10 hours is required. But in our body, the dilute HCl, the pH 1.2 to around 1.5 to 2, in that case, the pepsin, that chicken was digested within 2 to 3 hours. So how the faster the rate of reaction? It is only possible by the with the help of the enzymatic action. So enzyme is a very important, not only the digestion but also enzyme all the metabolic reaction, and every day more than 1000 types of reaction occurred with the help of enzymes. So in that case, thousand different types of reaction will be occurred every day for daily life, and each and every reaction with the help of the enzymes. Without enzyme, that cannot be possible. And simultaneously, all enzymes are the protein in natures except the ribonuclease. Ribonuclease is the nucleic acid derivative. So let us start. Enzyme is a biocatalyst. Enzyme are the substances, is a very important substances. Those are help for the catalytic reaction and all for the long life of the life process and continue the life process. And just like a teacher acts as a enzymes in between the student. When a student read some things 10 to 20 times, but when a teacher read this or understand these things or the topics, I think it will be reduced lesser the times. It is true or false? It is true. And suppose
Yes,
you
sir.
read one topics, you read one topics, one chapters. Let's suppose enzymes. You read in only in itself yourself you can study. Then the time is required. But when I study after teachers or I presentation my lecture, then you can study. There is a lesser the time. So your teacher just like a enzymes or just like a another catalytic action, just like a enzyme and the substrate and enzyme student teacher relation, just like a another biocatalyst. Okay, so this is very interesting topic, and these enzymes also help for the so many disease and disorder. So if it increase the enzymatic action, there is a one type of disease. If it is decrease the, there is another type of disorder. So any disease or disorder that can be identified by the enzymes with the help of estimation of the enzymes. So be careful, be listen it, seriously. If any sort of problems or any do not understand it, please ask me at that time, because this topic in your syllabus, this topic in your second slot examination. So be careful. Today's up to the class, it will be incorporated in your second slot examination. First, let us see that what is enzymes. The basic chemistry and history of the enzymes, classification, mechanism, enzyme kinetics, inhibition, activation, and the specificity. This is the basic chemistry of enzymes. Enzymes are the substances which are protein in nature, which are present in a particular concentration, which act on the specific substrate and on the specific pH, temperature, and the specific condition. This is called the enzymes. Enzymes are the biological catalysts that speed up the rate of reaction of the biomolecules. And that most enzymes are the three-dimensional. Another most important characteristic feature of the enzymes is it is the three-dimensional globular protein, tertiary or quaternary. Because these three-dimensional having, there is some binding site and there is some other site that is called the allostetic. Let us discuss elaborately. And some special RNA species also acts as enzymes. They are called the ribozymes, as per the ribozymes. This is the short history of the enzymes, how to discover these enzymes. The Berzelius in the 1836 coined the term catalysis. Actually, this enzyme terms is not previously, it is termed as a catalysis because it is catalysis the some rate of reaction. Catalysis the reaction. That's why catalysis to dissolve. That means the substrate can dissolve it. So previously enzyme is named as catalysis. In 1878, Kuheni used to word the enzyme first, and Greek word that means the yeast. This is very important, you can remember it. The enzyme means yeast, and this name was coined by the Kuheni. Kuheni 1878. Kuheni used the word Greek enzymes. That Greek word, it means the yeast, to indicate that catalysis takes place in the biological systems. Next, the isolation of the enzymes. First isolation of the enzymes from the system from cell-free extract of the yeast achieved by the 1883 by the Buchner. Buchner was the scientist who first extract the cell-free extract the enzymes from the yeast. Then he named the active principle of the zymase, and later found it in the mixture of the enzymes could convert in the sugar to the alcohol. This is known as the zymogen. Zymogen is the one of the enzyme which is helped for the convert the sugar into a alcohol. In the 1926, James Sooner first achieved the isolation and crystallization of the enzymes urea from the jack beans and identify the proteins. Okay, this is the short story. The James Sooner also get the Nobel Prize on 1946, and he was the first crystallized the enzyme urease. In the year 1930, John Northpole, he got the Nobel Prize. In the year 1946, crystallization in number of proteolytic enzymes from the gastrointestinal tract. Proteolytic enzyme means pepsin, trypsin, and the erepsin from the gastrointestinal tract, and proved that they are all are protein in nature. So especially enzymes are protein in nature, having a three-dimensional structures except the ribonuclease, but basically present in the cell for increase the rate of reaction. So they are acts as a biocatalyst. Now we can see what are the types of enzymes and what are the main component of the enzymes, how act on the substrate, and the structure of the enzymes. This is very important. First of all, the active side of the enzymes. Enzymes, what are the characteristics of the almost all enzymes of the protein in nature, they are heat level. Another is most important parameter. This is heat level. That means they are not heat thermostable in presence of heat, because enzymes are composed by the proteins. So protein, in presence of protein, they are denatured. So that's why this is very important. It is heat level, and the maximum activity of the enzymes is 37 degrees centigrade temperature. That's why our body temperature 37 degrees centigrade, because in that temperature enzyme is very active. That's why our normal body temperature is the 37 degrees centigrade. And this is also another important property. It is water soluble. It is water soluble in nature, and they can be precipitated by the protein precipitating reactant like ammonium sulfate, and heat, strong acid, strong alkali, tannic acid, and they contain 16% weight of the nitrogens. 16% weight of the nitrogens. The structure of the enzyme, first of all, it is a three-dimensional structure. It's a one is the active site, another is the allosteric site. Allos means other, Greek word. Allos means others, and active site means where the binding site of the substrate. Generally, the enzyme which bind with the substrate, that small portion of the site is called the active site, where the substrate can bind and where the drug also can bind, or where the receptor cannot bind. So these are the active site of the enzymes in the region that bind the substrate, cofactor, prosthetic group, and contain a residue that helps for hold the substrate. So there is a few terms coined here. One is the cofactors, another is the prosthetic group. Be serious. What is enzymes? Then the cofactors and the prosthetic groups. Active sites are generally occupy less than 5% of the total surface area. So it is a so small area of enzymes that will occupy only the 5% out of 95%. Most of the enzyme have the no allosteric site, but only the 5% is the active site where the drug can bind, where the substrate can bind, but which can be bind, that is dependent upon the enzyme specificity. This active site of the specific shape due to the tertiary structure of the protein. This active site, this side is the active side, is the tertiary site. Just like you purchase one pen having a 3 to 4 pockets, and you can kept 1 to 3 goods or things for the different pockets, and depend upon the size of the pockets. So that's like a enzymes have some pockets and bind with some substrate, and it is dependent on the specificity of the substrate. Some substrate bind with some specific enzymes. It is not true. All the substrate bind with the all types of enzymes. Only the some specific substrate can bind with the specific substrate. That is called the specificity of the enzymes. Another is change in the shape of the protein affect the shape of the active site and function of the enzyme. But this shape of the site, active site, does not fix. Sometimes it is flexible in nature. There is some little bit of conformational changes in the presence of the substrate or the drugs. There is some conformational changes, and they bind with the substrate, and finally the enzymatic action or catalysis will be done. To active site, it will be done by the binding site. Another is the catalytic site. It choose the substrate to bind the active sites, and catalytic site, it perform the catalytic action of the enzymes. That means active site first to bind the substrate, then catalysis occur. Then what is cofactors? Cofactor, write down this. The cofactor, it is the non-protein molecule which carries out the chemical molecule reaction that cannot be performed by the standard 20 amino acids. Cofactor are categorically two types, and the organic cofactor, another is the inorganic cofactor. And these cofactors divided into two types. One is the coenzymes, another is the prosthetic groups. Cofactors, this is the cofactors, and this is the inactive protein. That means inactive protein which is cofactor, but the active protein which binding L1 and L2, this is the cofactor activated the protein. So cofactor, one of the most important parameters that can bind with the active side of the enzymes, then the enzyme action will be done. So without cofactors, enzyme action never be done. Say for example, in case of glucose, in case of glycolysis, glucose is converted to glucose 6-phosphate in the presence of hexokinase or the glucokinase. But this glucokinase or hexokinase activity never possible if the magnesium cofactor or coenzyme is not present in the solution. So magnesium acts as a cofactor. Without the coenzyme, enzymatic action is totally inhibited, or enzyme action is not possible with the help of coenzymes. So inorganic cofactors, these are the inorganic molecule required for the proper activity of the enzymes. Enzyme, carbonic anhydrase, zinc is the cofactors activity. Hexokinase for the magnesium, already discussed. And organic cofactors, they are the organic molecule required for the proper activity of the enzymes. Glycogen phosphorylase require the small organic molecule pyridoxal pyrophosphate. And the types of cofactors, one is the prosthetic groups, another is the coenzymes. Type of cofactor, one is the prosthetic groups, another is the coenzyme. What is prosthetic groups? Prosthetic groups and the coenzyme, their example are the same, but in the difference, when the cofactors on the or the element which tightly bound with the enzymes, they are not easily separated by the simple dialysis, that is called the co-prosthetic groups. An example of prosthetic group is the vitamin B complex group of thiamine, riboflavin, basically the thiamine pyrophosphate, FAD, NAD, FADH2, all are the prosthetic groups. And simultaneously, sodium, magnesium, copper, manganese, zinc, all are the prosthetic groups. But simultaneously, these example also the coenzymes. But what is coenzymes? Coenzyme are the non-protein part of the enzymes which are loosely bound with the active sites. Loosely bound with the active site, that is called the coenzymes, and prosthetic groups which is tightly bound. That is the main difference between the prosthetic group and the coenzymes. Where if the compound tightly bound, which are do not separate by simple dialysis, that is called the prosthetic group. But the example are the same, and same compounds, sometimes it is tightly bound or sometimes it is loosely bound. When it is tightly bound, that is called the prosthetic group, and when it is loosely bound, that is called coenzymes. But compound are the same. But why when it is tightly bound and when it is loosely bound, it is depend upon the enzyme and the coenzymes or the prosthetic groups. Some enzymes, some coenzymes, some agents which bound with the tightly of the different enzymes, and some elements which are bound with the loosely with this. So that is called the coenzymes. So this enzyme which is the cofactor remove the designated as a apoenzymes. Apoenzyme means actually enzyme is the hollow enzymes. Hollow enzymes. Enzyme having two parts. The whole enzyme is called the hollow enzymes. It contains one protein part, another is the prosthetic or cofactor. That is called the this protein part of the enzyme, it is called the apoenzymes, and this another part is called the cofactors. This complex, the complete complex of the protein will be necessary small organic molecule, metal or other component is termed as a hollow enzyme or hollow proteins. So these are the active side, these are the enzyme, the protein part of this compound, it is called apoenzymes, and this is called the active side. It is a small portion of the enzymes, this is active enzyme, and there is some protein channel or it is called the apoprotein, and there is some cofactors which bind with the enzymes. This is the cofactor, that is called the prosthetic groups. So protein part is the enzyme is called the apoenzymes and bind with the cofactors, it is called hollow enzymes বা whole enzymes. Whole enzyme consists of apoenzyme plus coenzymes. Okay, any question up to this? Hello, any question?
No, sir.
Do you understand what is a?
Yes, sir.
Okay, any other students? Because this is very important. today's class is incorporated in your second slot examination, as well as it is help for the so many types of disease disorders and the digestion metabolism. So your digestive system, what type of enzymes act on the which manner and which the specific substrate can. So this chapter is very important. I think both the section A and section B, I think this topic is covered by me. Okay, this is enzyme because I think this will be covered you, then I start the metabolism. Otherwise metabolism cannot be start without study of enzymes. Enzyme is such a substances that can help for the digestion or for the metabolism. Any chemical reaction without enzymes cannot be possible, and simultaneously enzymes along with the coenzymes. And what are the, and these coenzymes, it will be classified actually this cofactor. Broadly it will be classified cofactors plus apoenzyme equal to holoenzymes. Write down this: cofactors plus apoenzyme is called holoenzymes. Holoenzyme means enzymes. It consists of two parts. One is the protein part, another is the non-protein part. The non-protein part either organic molecules like vitamin B complexes or the metal ion. That is called the cofactors. So this non-protein part is called the cofactors and protein part is called the apoenzymes. And this cofactor again classified into two groups. One is the prosthetic groups, another is the coenzymes. Another is the coenzymes. That means when the substance is tightly bound with the enzymes, that is called the prosthetic groups. Never it will be separated by the simple dialysis by any other method. But when it is loosely bound, that is called the coenzymes. An example is the same: the organic compounds vitamin B, FMN, riboflavin, FAD, NAD, NADPH, ATP, GTP, all are the coenzymes, but depend on the tightly bound. Just like a magnesium stearate which are used in the pharmaceutics. The magnesium stearate, sometimes it is called the lubricant and sometimes it is called the glidant. When the magnesium stearate increase the flow property, that is called glidant, and when the magnesium stearate it reduce the friction of the hopper and the granules, that is called the lubricant. So example is the same, but when their activity, their name is different. Magnesium stearate sometimes acts as a glidant and magnesium stearate sometimes it is as a lubricant. Here also same, the riboflavin, suppose FMN, flavin adenine dinucleotide, which is FAD, which is as a prosthetic group, when it is tightly bound with the particular enzymes, and when it is loosely bound with this, it is called coenzymes. I think clear?
Yes, sir.
Yes, sir.
No problem. Thank you. The next to the gloss and go to the next slide. Next, substrate. Actually, main function of the enzyme is to bind with the substrate and to catalysis the substrate. And this is our main method. We are taking some foods or nutrients. This is acts as a substrate. And this substrate, when goes to the stomach or goes to the orally or by any means, this enzyme bind with the substrate and this catalysis will done. Suppose you take starch, carbohydrate or rice. This is the polysaccharide. When bind with the amylase, amylolytic enzyme, the amylase which is digest or to break down this product to a smaller molecule and which is converted into a glucose and which is absorbed. So this is the reactant in the biochemical reaction is termed the substrate, and when a substrate bind to the enzymes in the form of enzyme substrate complex, this is the enzyme substrate complex, but it is very specific in nature. A particular substrate can bind with the particular enzymes. That means carbohydrate which is bind with the amylolytic enzyme, not in the proteolytic or lipolytic enzymes. Simultaneously, Proteins can bind with the pepsin, trypsin, erepsin, not in the aminolytic or lipolytic enzyme. Lipid can bind with the lipase, or lipase can bind with the lipid, not lipid bind with the amylase or proteolytic enzymes. So enzyme substrate complex will be formed a very specific manner. It will be very specific. Particular substance bind with the particular enzyme, just like a particular key open a particular lock. Any other key, sometimes it will be matched, but generally a particular key open a particular lock. Substrates are the particular substance can bind with the particular substrate. That is called the enzyme specificity, because all enzyme is very specific. Proteolytic enzyme only digest the protein. Amylolytic enzymes which digest the carbohydrate, and lipolytic lipase only one enzymes lipase which is catalyzes the only lipid. So they are very specific. This is not true. All enzymes act on all the substrate. This is not true. The site of enzyme synthesis. The enzyme synthesis in the ribosome, which is attached to the rough endoplasmic reticulum, ERP, and formation of the synthesis of carriers on the DNA and an amino acid bonded together to form a specific enzyme. Actually, better enzymes are the two types. One is the intracellular enzymes, another is the extracellular enzymes. Inside the cell, another is outside the cell. The extracellular enzymes, another is the intracellular enzymes. Intracellular enzymes are synthesized or written in the cell for the use of cell itself, and they are found in the cytoplasm, nucleus, mitochondria, and the chloroplast. Example: oxidoreductase catalyze the biological oxidation. Enzymes involved in the reduction in the mitochondria. In extracellular enzyme, those enzymes which are synthesized in the outside the cell and secreted from the cell work to extremely digestive enzymes. All the digestive enzymes, pepsin, trypsin, erepsin, amylase, lipase, all are the outside the cell. But in case of glycolysis, in case of TCA cycle, in case of beta oxidation, that means all the enzymatic action in the inside the cell, all are intracellular. And extracellular, that means outside the cell, which act on the outside the cell, especially for the digestive enzyme, which act on the extracellular enzyme to digest the protein, carbohydrate. What are the characteristics? Enzymes, first, speed up the reaction. Then obviously the lowering the activation of the the lowering the activation of energy. And their process, their presence does not affect the nature and property of the end product. Their presence, the presence of the enzyme does not affect the nature of the reaction and the product. Producter কোনো রকম problem change হয় না. And they are highly specific, already discussed. They are highly specific. One enzymes act on the same type of substance, not the others. Their action, there may be enzymes and be catalyzed on the kind of substrate. A small amount of enzyme can accelerate the chemical reaction. A small amount of enzymes can accelerate the chemical reaction. বললাম তো 100 times, 10 to 100 times folds increase the rate of reaction. Just a concentrated hydrochloric acid that does not digest the piece of chicken within a 8 to 10 hours, but a few hours, within 2 hours, a chicken will be digested in the presence of the pepsin or enzymes. Enzymes are very sensitive because to the pH. It is very much pH dependent. Also the temperature and the light also. Basically the pH and temperature. That's why our body temperature 37 degree. Most of the enzyme, their optical activity, most of the enzyme, their optimum temperature is the 37 degree centigrade. That's why they are well acted on the body at the 37 hours. That's why our body temperature 37 degree centigrade and the pH specific. Some enzymes, pepsin, which are act on the acidic pH, trypsin and erepsin, which act on the basic pH. Turnover number, this is the defined as the number of substrate molecule transformed per minute by the enzymes molecule. And the substrate will be changed or turnover. Number of substrate molecules turnover per mole of enzymes. That is called the catalyzed turnover number. It is equal to 6 into 106 per minute. The minute per one, that is called the turnover number. The nomenclature of the enzymes. This is very important. What are the nomenclature of the enzymes? That means everything, every substance having some names. Your name, for 50 student, 50 can all of the whole of the world, they are having some specific name to identify it, and also some genus and species. But how to nomenclature? What are the name of the, what are the naming system of the enzymes? And enzymes is named according to the name of the substrate catalysis. Some enzymes are named before a systematic way naming system, like pepsin, trypsin, and erexin. This is called the arbitrary name. Yes, this is called the arbitrary name. There is a pepsin, trypsin, some small enzymes are invented and they are act on the different substrate, and that type of naming system is called the arbitrary name. Next, this is the problematic when the other enzymes come, then it is adding AAC at the end of the name of the substrate. Suppose last class I discussed about the maltose is the substrate, enzyme maltase, AAC maltase is the substrate, and AAC is suffix maltase, sucrose, sucrase, lactose, lactase. So AAC is the suffix just based on the name of the substrate, just name of the substrate, and AAC adding with the AAC. This is called the trivial name. This is called the, first is the arbitrary name, then the other enzymes come, then substrate, the which enzyme act on the which substrate, that is called substrate specific, and only the name of the substrate plus AAC equal to name of the enzymes. Name of the substrate plus AAC equal to name of the enzymes, maltase, lactase, sucrase, so many substances. But there is some problem. The maltase plus water, maltase, glucose plus glucose. This is called the trivial name. Mane kore rakho, write down this. This is called trivial name. First one is the arbitrary name. যেরকম তুমি জন্মানোর পরে একটা আরবিটারি নাম দিয়ে দেওয়া হয়, তারপরে আবার তোমার অ্যাক্টিভিটির উপরে ডিপেন্ড করে আবার কেউ কেউ নাম দেয়, নাম হয়ে যায় একটা, আবার কেউ একটা অফিসিয়াল নাম থাকে, সেটা তোমার বই, স্কুল, কলেজ, মানে সব জায়গায় নাম দেওয়া থাকে। তো এখানেও প্রথমে ইনিশিয়ালি কিছু এনজাইম এসছে, জাস্ট আরবিটারি নাম দিয়ে দেওয়া হয়েছিল, পরে দেখলো না এটা দেয়ার ইজ সাম প্রবলেমস, হাউ টু ডিফারেন্সিয়েট দিস নেমিং সিস্টেম। যেরকম তোমাদের ফোনে দেখো বিভিন্ন সময় আগে নাম ছিল, বিএসএনএল এর একটা নাম্বার দেখলে বোঝা যেত এটা বিএসএনএল, জিও দেখলে জিও, এয়ারটেল দেখলে, কিন্তু নাওয়াডেজ ইট ইজ কোয়াইট সিস্টেমেটিক ওয়েতে করা হচ্ছে যাতে বোঝা যায়। তো দিস ইজ কল্ড দ্য ট্রাইভাল নেম। তাহলে ফার্স্ট ওয়ান ইজ দ্য আরবিটারি নেম, সেকেন্ড ওয়ান ইজ দ্য arbitrary tribal name, but there is a problem occurred. One enzyme name is the catalase. Enzyme name is the catalase. So catalase is the enzyme's main functionality to catalyze the substrate. So which substance can be catalyzed? That is very difficult to identify the which substrate and which category of the enzymes. So that's will the problem arises when the catalase will come and discover it. Then the scientists, this is the maltase, lactase, sulfase, lipid, lipase, starch, amylase, and amylopectase. Then problem will become. Then how will we overcome these problems? Then a commission will be framed, that is called the International Union of Biochemistry and the Molecular Biology. International Union of Biochemistry and Molecular Biology, IUBMB, in the year 1964, and modified in the 1972 and 1978 is suggested the IUBM, the systematic nomenclature of the enzymes. It complex that and compresses the ambias as per the system. They are starting at the EC now. Actually, they have formed some EC, enzyme commission. That enzyme commission produce a 4-digit number, EC 4-digit number, and EC 4-digit number. And in this methodology, they are having this all the enzymes classified into a 6 major classes. Or that is called the International Enzyme Commission. They say there should be the 6 major classes: oxidoreductase, oxidoreductase, transferase, hydrolase, lyase, isomerase, and ligase. The name and the position are very much fixed. These are the name of system: oxidoreductase, that means oxidation reduction; lactate dehydrogenase; transferase, move the chemical group hexokinase; hydrolase, for hydrolysis, bond cleavage and transfer; lysozymes; and the lyase, non-hydrolytic bond cleavages, that is fumarase; and isomerase, materially through isomerization reaction, glucose to fructose, fructose to glucose. The intramolecular group transfer, that is called the isomerization, triphosphate isomerase, and ligase, the RNA polymerase. So these are the 6 major classes. These are the 6 major classes এবং their position. 1, that means oxidoreductase; 2 means transferase; 3 means hydrolase. Suppose EC 1, 2, 3, 4. That means first one is the major class, second one is the subclass, third one is the subclass, fourth one is the subclass, and that will be actually first class, মানে first digit 1, first digit which is 1 to 6, second digit 1 to 100, third digit 1 to 1000, and fourth digit 1 to 10,000. So you can remember only the first digit. EC ধরো 1111, it is a lactate dehydrogenase, but 1, that means oxidoreductase. This enzyme number and the name is very much fixed. এরকম নয় জাস্ট ভুলে গেছো লিগাজেস প্রথমে লিখলে নাম্বার 1 লিগাজেস, নাম্বার 5 আইসোমারেজ, নাম্বার 2 আইসোমারেজ, এরকম হবে না কিন্তু। দের যে this number and position is very much fixed. 1 first oxidoreductase, and under this subclass another 3 classes, but that will be 1 to 100, 1 to 1000, 1 to 10. That will be not remember you, that is very difficult. It will be go through the internet, you can go through the what is the naming system, what are the 1 to 100, 1 to 1000, all are given in the internet. So this is not possible. Just you can remember the first digit of the number, or suppose EC 234, 234, that means the enzyme is 2 মানে transferase. So 378, so the only you can remember 3 means hydrolase. Okay, so up to this nomenclature classification,
হ্যালো স্যার।
up to this okay, time শেষ নাকি?
না স্যার, বলছি ওই লাইসেস যেটা আছে লাইয়েস হবে না ওটা?
লায়াজেস, এইটা ঠিকই আছে
স্যার।
এর
ওয়াইয়ের পরে যে এসটা আছে,
কি বলে
স্যার ওয়াইয়ের পরে যে এসটা আছে ওটা হবে?
হ্যাঁ, ওটা হবে না, দাঁড়া ওটা কেটে দিচ্ছি।
ইয়েস,
থ্যাংক ইউ। লায়াজেস and ligases. Ligases means cleavage without adding water, মানে cleavage মানে বন্ড ক্লিভেজ করার জন্য লাইজেশন, এন্ড ligation মানে জয়েনিং করার, যে দুটো বন্ডের মধ্যে যদি জয়েনিং করে দেবেন পলিমারেজ ফিউমারেজ দুটোর মধ্যে ব্রেকডাউন, দ্যাট ইজ কল্ড দ্য লাইজেস, এন্ড লিগাজেস হচ্ছে লাইগেশন করে জুড়ে দেওয়া, দ্যাট ইজ কল্ড দ্য ligases. So up to this nomenclature in your second short examination, first epimerization, then anomerization, mutarotation, and the enzyme, and then nomenclature of the enzymes. Enzymes, brief history of enzymes, and introduction to enzymes along with nomenclatures. So I think, do you understand? If any question, please ask me.